Science in Pajamas. I know it's been a little while since I've done one for, um, actual science as opposed to just the wellness ones. So today I was going to talk a little bit about something called epigenetics. Now, epigenetics is when there is a change to the expression of a gene. Now, normally when we see a change to the gene expression, it's because there's a change to the DNA, which is a mutation. But epigenetics is when the expression changes, but not the DNA. There's actually zero change to the DNA. Now, how this works is because of certain chemical groups that can actually attach to the DNA on the outside and can affect whether or not they get transcribed. So here I just kind of have a breakdown of, what your DNA looks like. Humans have 46 chromosomes, 23 pairs, and a lot of times we show a chromosome like this, where we have the centromere, the proteins that keep the sister chromatids together. But what the chromosome actually is supercoils of the genetic material of the DNA. And if we take those supercoils and expand them out, we see these coils of these things called nucleosomes. Now, what a nucleosome is, if we expand that out, it is a bunch of histones and DNA that are wrapped up together. Now, what does that mean? Well, histones are particular proteins that the DNA will actually wrap around. And then if you have a whole bunch of histones with the DNA wrapped around it, then they can coil up and wrap around each other, and that's what the nucleosome is. And as we said, what's wrapped around the histone? The actual double helix of the DNA. This is one way that, this is pretty much one of the big explanations of why we can fit so much genetic material into such a small area like our nucleus. It's because it is wound up again, coiled, supercoiled, and that's how we get our chromosomes. So we take the DNA, we wrap it around the nucleosomes. We take these histone complexes, multiple of them, and we wrap those up. That's how we get the nucleosomes. And we take all these nucleosomes and we coil them up together. And then we take those coils and we coil them even more. And that's how we get our chromosomes. Now, that's very important for when we talk about epigenetics. So epigenetics is when certain chemical groups are added to the outside of the DNA, and they can actually affect whether or not a gene gets expressed. What I mean by that is, if you remember, we talked about the central dogma. The DNA gets transcribed into RNA, specifically messenger RNA. So it gets transcribed into mRNA. It goes out into the cytoplasm, and it gets translated into the protein. Bear with my bad writing. I am kind of writing a little bit at a side, or sideways. So it's this step right here that is super important, because you cannot you can't express a gene if you don't have the mRNA to translate. So what happens is the human genome, and genomes in general, are pretty freaking amazing. Not all genes are expressed all at the same time. As you grow and you age, different genes get turned on and off. I'm not even just talking about how certain genes are being expressed in your skin and certain other genes are being expressed in your brain, and this is not the same set of genes. But even your blood, we actually make 3 different forms of hemoglobin. We make one, which is embryonic hemoglobin, and it kind of helps, before we really get into the development, it just helps move oxygen and nutrients around that wonderful glob of cells that is us. And then we have fetal hemoglobin, which has a different structure. And it's more designed to start carrying the blood through the rudimentary circulatory system that we are forming at that time. And specifically, it's designed to pick up oxygen from the umbilical cord as opposed to the lungs. It doesn't pick up oxygen from the lungs because while you're still in development, while your mother is still pregnant with you, you're not breathing on your own. And then once you are born, the genes for the fetal hemoglobin turn off and the genes for the adult hemoglobin turn on. And that's the structural version of hemoglobin that will pick up the oxygen from the lungs, deposit throughout your body, pick up the carbon dioxide, deposit that in your lungs, and keep going like that. So we have different proteins and enzymes that will regulate gene expression. When should it be expressed? When should it be silent, meaning not expressed? But sometimes we can actually change that expression. Now, we always have these chemical groups on our DNA. They're what control gene expression as it is. So there are three main ones, although two of them are the ones we're going to focus on here and in class. So we have methyl groups, we have acetyl groups, and we have phosphate groups. Now we're going to focus mostly on methyl and acetyl, and we're not really going to mention a whole lot about phosphates. They kind of do a little bit of both of the jobs of these, but only under certain circumstances. Now when a methyl group is added to the outside of the DNA, so we mentioned that you have your, where's my green? So we have the histone, and then we have the DNA wrapped around it. We have it wrapped around it. And then what can happen is if a methyl group gets added onto the outside, so it's just added directly to the outside of the sugar phosphate backbone. If a methyl group gets added to it, it's going to block that gene from being transcribed. So right now in our bodies, we have the adult hemoglobin version of hemoglobin structure turned on. The gene for the fetal and embryonic hemoglobin, those genes have methyl groups on them, and that is what prevents them from being expressed at this point. And the way that it works is when the methyl group attaches to the DNA, it actually contorts the DNA, causing it to wrap even tighter up around the histone. So since it's wrapped even tighter up, those transcription factors cannot access it, which means that it's not going to be transcribed. And if it's not transcribed, we can't translate it. So right now, the genes for the fetal and embryonic hemoglobin have methyl groups on them, which is causing them to tighten up even more tightly around the histone, blocking transcription. And right now, what we do have, though, we have acetyl groups attached to the DNA where the gene for the adult hemoglobin is. When the acetyl groups are added, it actually contorts the DNA so it loosens up a bit around the histone. Now that it's a little looser, all the transcription factors can squeeze in there, have access, they can transcribe that gene, and since it's been transcribed, it can now be translated and we can make the protein from it. So the acetyl group loosens up the DNA and allows it to be transcribed. So our gene for adult hemoglobin now has been acetylated. It has an acetyl group on the outside, which is why it's so easy for us to transcribe it and thus make the protein, the adult hemoglobin. So this happens normally. This is going on all the time in all of your cells. I mean, you don't want your skin cell making hemoglobin, so in the like skin cells, they're not going to be making blood cells. Only your bone marrow makes the blood cells. So actually, all of the hemoglobin genes, embryonic, fetal, and adult, all of those are silenced in your skin cells, meaning they're pretty much all methylated, because you don't want your skin to do that. You don't want it to start oozing blood and oozing hemoglobin. Not exactly a good thing. But the cells that we do have that are making blood, our red blood cells, and making hemoglobin for those cells, those are the ones that have it acetylated. So it has the acetyl group attached to it. So this happens normally throughout our life, from the moment we are conceived to the moment we die, and probably a little bit after that, because your cells do live a little bit after you have officially died. Our cells are naturally controlling gene expression. But with the idea of epigenetics is that it's not just regulated by our cells, that different things can also cause our DNA to be methylated or acetylated beyond our normal genetic control. And this can have different kinds of effects or disorders, or lead to higher cases of certain characteristics. For instance, in April of 1945, we have what's called the Dutch Hunger Winter. It was February through April. And what happened is, if you know your history, this is around the time of World War II. And the Nazis had actually blocked all flow of supplies, especially food, going into different cities in parts of Holland. As a result, malnutrition, starvation was rampant in those areas. What they found, though, is that it wasn't just that people were suffering from malnutrition, but children who were alive and survived that time actually grew up, and they noticed a spike in instances of schizophrenia. Now, they looked into this later, and what they found is that the stress of the malnutrition actually prevented certain proteins from being created. So specifically IGF-2, I believe, so that's insulin-like growth factor. And this is a particular growth factor that helps with cognitive abilities such as thinking, perception, and as a result, since that was hindered, that particular gene had been methylated in these individuals, so they lacked the IGF-2, and as a result, when they were older, since they lacked the expression of that gene, it affected their perception and led to higher cases of schizophrenia. Now, people who were born after that, or even before that, but were already grown before that, or children who were born after that, since they didn't go through that particular situation, they weren't malnourished, their genes for the IGF-2 didn't get methylated, and they had normal development and a much less incidence rate of schizophrenia. However, the interesting thing about epigenetics is, even though it doesn't affect your actual DNA in terms of the bases, it can still be passed on. So those who did have this higher incidence of schizophrenia, their children had a higher chance of getting schizophrenia too, because the methylation in the parents' genes was still prevalent in their offspring. So it can be inherited. However, the thing about epigenetics, yes, they can be inherited, but they can also be reversed, and that's what they found, is that over time, as other situations arose, bountiful harvest and things like that, those genes that prevent, or the methyl group that prevented the IGF-2 gene from being expressed, eventually the methyl group came off, the DNA loosened up enough that transcription can occur, and then now that they had adequate nutrition and all that, the next generation, they did see a lower rate of schizophrenia. So it's just kind of something interesting about epigenetics, how it changes the expression of your genes, but it doesn't actually change your DNA bases. How it can be inherited, but it can also be reversed and then no longer inherited. So that's just something I wanted to mention to kind of hopefully help us out a little bit understanding epigenetics. I hope you found this interesting. I hope you found it informative. And again, if you have questions, hit me up in our Google Classroom, or guys can email me. So best of luck. Hope everyone's staying healthy and safe. And I will see you in our virtual class. All right. Bye-bye.